Here we go, component 2 in class question book, and question 9 we're on to inheritance and genetics. A bit of a monster question this, so chance to shine for 17 marks. A lot of reading and looking at diagrams. In mice, coat colour is determined by genes at more than one locus. And so the locus is just the position of a gene on a chromosome. And we've got locus 1, and that's the colour with two alleles, big B and little b. Big B for black, little b for brown. And locus 2, the distribution of the pigment. We've got big A, and that will produce agouti with the bands, yellow bands on a dark background. And little a is the non-agouti, where it's no yellow bands, just the dark colour. So that's a little bit of biology, and then we've got a table telling us exactly what will happen for our genotypes and phenotypes. So all we need to do is follow some rules to complete a genetic diagram. The expected outcome of crossing mice heterozygous at both loci. So heterozygous, the alleles are different to each other and both loci. So that's pretty straightforward to start with. So heterozygous, we're going to go big B, little b, big A, little a. So remember our golden rules. We have in a normal individual, for normal cells, we've got 2 copies of each gene. So 2 copies. So an individual must have 2 copies of B and 2 copies of A. And we're crossing mice heterozygous for both. So the other one is also big B, little b, big A, little a. And that is the genotype, which I've just written in the phenotype box. So I will just transfer those down. And phenotype, so it's alright to make mistakes. I'm just going to put a single line through it and write the correct answer after it. No fuss. And the parental phenotype for if you're just looking at our table, we've got our top row here where we can find our big B, little b and our big A, little a, and that phenotype. So we're just reading the data from the table, and that says normal agouti. So I'm just going to copy that out. Normal agouti and normal agouti. Gametes. So in choosing gametes, we need to remember meiosis, and meiosis produces haploid gametes. There's a reduction division, so we're just having 1 copy of each gene. But what could we have here? Well, we could have either of those 2 B's, and we could have either of those 2 A's, and so we've got a range of possible gametes that we could have. So we could have a big B and a big A. We could have, so I'm going to put a comma to separate these, a big B and a little b and a big A, and a little b and a little a. So all of those gametes are possible. And you know about independent assortment in meiosis, and that makes those possible. And the other one is exactly the same. So big A, big B, A, big B, little a, little b, big A, little b, little a. And complete a genetic diagram. So I'm going to draw the standard Punnett square. I used to think was named because it looked like a strawberry punnet, but actually it is the scientist Punnett But you won't catch me admitting that on video.
I wonder if his first name is Strawberry.
Strawberry Punnett. Anyway, all Punnett squares look the same. They're a box that looks a bit like a strawberry Punnett box. So we're just going to draw a box. And we're going to have our gametes in 4 spaces that are available. Okay, so that's what Punnett squares look like, and we've got the male and the female, although it doesn't really matter in this case. So we're just going to transfer our gametes into these headings. So big B, big A, big B, little a, little b, big A, little b, little a. And we're going to do the same down the side. Big B, big A, big B, little a, little b, big A, little b, little a. And it is rather confusing in this example. You want to put the A's before the B's, don't you? That's a sensible way of doing it, but they've tried to make it just that little bit difficult for you, because that's the sort of thing they enjoy doing. And so now we've just got our straightforward adding up process. So we just add these all together. You're generally quite good at doing these when you do these in class. So we're just adding these up, a little bit tedious but easy to make mistakes. So big B's, and then we've got big A, little a, and the convention of writing these is to put the capitals first. So big B, little b, big A, big B, little b, big A, little a. 2 big B's, big A, little a, 2 big B's, 2 little a's, big B, little b, big A, little a, big B, little b, 2 little a's, big B, little b, 2 big A's, big B, little b, big A, little a, 2 little b's, 2 big A's, 2 little b's, big A, little a, big B, little b, big A, little a, big B, little b, 2 little a's, 2 little b's, big A, little a, 2 little b's, 2 little a's. So I've just added those gametes together, and that's what would happen when they fused with each other. And then we are giving the ratio of the following phenotypes. So we need to just, I actually write symbols or letters in the box to associate the phenotypes to the genotypes. So anything that is at least heterozygous for both, so either big B and little b, and big A, little a, so for both, if there's a dominant allele for both of those genes, then it's going to be normal agouti. So anything that's got a capital for A and B is normal. So I'm going to put N-N-A, so normal agouti. For anything here, that one's normal, 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 normal. That one isn't, it's got 2 small a's. That one is 2 small a's, and that one's normal. My studio is falling to bits. There's feedback from everyone there. Not too many shadows on the paper. Not now with our new lighting situation. It's Biz Recall show. And 2 little b's. That's not going to be 2 little b's. So that one's going to be normal agouti. And the rest have got so 1, 2, 3, 4, 5, 6, 7, 8, 9. So counting those, we've got 9 that are normal agouti. Solid black. So anything that's solid black has got to have 2 little a's and then at least a dominant B. So 2 little a's, there we go, and the solid B. So that one is going to be black. I'm going to put that in a circle, make it stand out. So 2 little a's, and so that'll be there, okay. And we've got anything else with 2 little a's and a dominant B. There we are, black there, and none others follow those rules. So we've got 3 of those. Then cinnamon. Cinnamon needs to be homozygous for B's, so 2 little B's, and then it's got to have a dominant A there. So homozygous would be the dominant A, so that's going to be cinnamon, so I'm going to use a C there. And this is 2 little B's and a big A, so that's going to be cinnamon. 2 little B's and A, that's going to be cinnamon as well, so that's going to be 3 of those. And the last one, 2 little A's, 2 little B's, and that's going to be solid brown. And so that's our 1 there. So if you do that enough times, you tend to get them right again and again. Over the page. A series of crosses between mice which were heterozygous at both loci, so that's the one that we did, produced 32 offspring, and the results are shown in the table below. A null hypothesis was proposed that there was no significant difference between the observed and the expected. That is the standard null hypothesis that you'd expect for this chi-squared. Using the table provided, calculate chi-squared. They have done a usual trick of not quite giving you enough table to do what you need to do. Although you can do it without the table, but it's a little bit mean, as we will see. So we've got observed, we've got expected, and then the next thing will be the observed minus expected, and I'm expecting, from remembering doing that table, so it's O minus E, and we square that. And then I know from my revision and my practice that there's another column that we usually put in, which is O minus E squared divided by E. So that's what we'd normally put in. Anyway, let's see what we'd expect. So if the numbers are quite handy here. There's 32 offspring, and let's go back to our ratio tables here. 9331. Well, that total is 16. That adds up to 16. So if we had 16 of these individuals, we'd expect on average 9 of them to be normal agouti, for example. And so if we've got 32 individuals, then we would expect twice as many of each of these 9331. So the numbers are quite handy. So for our normal agouti, so this was our 9331. So I'd expect twice as many. So I'd expect 18 of those. I'd expect 6 of those, 6 of those, and 2 of those. And quite often in these exam questions, the total number of individuals is some multiple of what you've got in the ratios. And then we just need to do a little bit of maths. So O minus E, so 22 minus 18, that is 4. 4 minus 6 is minus 2. 3 minus 6 is minus 3. And 3 minus 2 is 1. And then we've got squaring these. So 4 squared, 16. Minus 2 squared, well, we get rid of the minus when we square it, so that's just 4 now. Minus 3 squared is 9. So it doesn't matter whether it's minus 3 or plus 3, the squared would still be 9, and 1 squared is still 1. And then we've got the last little step in our normal table. So I'll make that table there, which is that number divided by our expected again. So we got, there's my trusty calculator, so 16 divided by 18. 16 divided by 18, which is 0.88888, which I'm just going to I'll call that 0.89. If the numbers are really tight later on, we can always come back and use more decimal places, but they're usually not. 4 minus 6, sorry, 4 divided by 6. So hang on, 4 divided by 6. 4 divided by 6. So that is 0.6 recurring. 6, 7, 9. 9 divided by 6. 1.5. And 1 divided by 2 is 0.5. And then this term here, which is adding them all up, is the final step. So we're going to add all of these up. So 0.89 plus 0.67 plus 1.5 plus 0.5 is 3.56. So chi-squared is 3.56. Bear in mind that actually in this margin on the day, the examiner won't be able to see that in the margin, so you might have to put it underneath or something like that. Anyway, that's our chi-squared. Choose an appropriate probability level and circle a critical value for chi-squared in the table below. So our probability is our we use the same level each time, which is 0.05, which is our 95% confidence or 5% error, or kind of 1 in 20. That's the standard level for student biology experiments. So 1 in 20 or 95% confidence. Our degrees of freedom is we take our number of classes that we've got and minus 1, so n minus 1. So we've got 4 classes, and so we're going to use 3. And where they join together in the middle, that's our magic number. What does it ask us to do? It asks us to circle it. So there we are. So that's 7.82. And now we're going to state whether we accept or reject our, remember this is a null hypothesis. So if you can remember, there's different ways of remembering this, but I always go back to if our critical value is greater than our, if our chi-squared is greater than our critical value, we can reject the null hypothesis. So if our chi-squared, 3.56, is greater than our critical value, 7.82, then we can reject the null hypothesis. It's not, so we must accept it. So I'm going to say accept the null hypothesis. Now, because my chi-squared 3.56 is less than my critical value of 7.82. Another sort of way of expressing that is that the probability of my chi-squared is now greater than our 0.05. You can see if we found it on this table, it would be here, it would be between 0.2 and 0.5, which is not good enough for our normal statistical way of thinking. And I'm going to take a break on that and then do a part 2, I think, because that's 20 minutes long.